Green here, okay. So this is gonna be the replacement for the suppressor screens, suppressor genetics lecture. So everybody will have access to this. But basically what we want to look at is some of the early work that geneticists did to try to get information on proteins and genes and how they function. The basic screening idea, a lot of the old timers, if you will, started with forward genetics, where you would just do random mutations using typically mutagens like EMS, ethane methane methyl sulfonate, or UV radiation or X-rays or something like that. Beetle and Tatum used X-rays, where you would just make random mutations in the genome, and then you would basically look for organisms that had a phenotype of interest. So think of like Thomas Hunt Morgan with his white-eyed flies. He got those through mutations. Or Beetle and Tatum made mutations in bread mold to find nutritional mutants. Or you can think of cell cycle mutants. People would mutate Saccharomyces to find phenotypes of interest. And then you'd have to do the hard work of sequencing or mapping or whatever combination of that you could do to try to figure out what mutations you made and in what genes they were made. So that's kind of where you start with a phenotype, and then you try to find the genes that are associated with that. Reverse genetics is the other way around, is where you target a known gene. And, nowadays you could do this with CRISPR. You could knock out a gene if you know the sequence, and then ask the question, did I get a phenotype from that? And does it have any effect that I would think would be associated with that gene? Or you can use RNA interference. So we learned about RNA interference. There you could knock down, so a CRISPR would be a knockout. You can knock down expression of a bunch of genes or one gene at a time, and then ask the question, what effect do I get from knocking those down, and even do combinations of genes. So the whole idea here is you're trying to screen kind of high throughput, trying to screen multiple genes at a time to try to get an idea of what kind of effect you're going to see from those mutations. Now, suppressor and enhancer screens kind of go along with these. Sometimes you mutate a gene and you don't see a phenotype. Either way, either through forward or reverse genetics. So sometimes it's nice to do these other screens, and you can typically pick up mutations in other genes that may not show a phenotype by themselves. They may only show a phenotype when combined with other genes. So two typical ways to do this, a suppressor screen and an enhancer screen. They pick up different types of mutants. A suppressor screen actually starts with a mutant phenotype, and then you try to suppress, either knock out or knock down genes that would suppress that mutant phenotype to bring it to more of a more wild type phenotype. Not all the time does it go all the way back. An enhancer screen, that's where you make multiple mutations, particularly in other pathways or processes, and then that would actually enhance or exacerbate those mutations, those mutant phenotypes. What kind of mutations can we get from these types of suppressor and enhancer screens? Well, you could think, if I do one mutation and that messes up the function of one protein, I could actually make mutations in other proteins that could compensate for that. So if you think about two proteins coming together to form a relationship or bind together, whatever, you could actually have two compensating mutations to do that. You can find gain of function or loss of function mutations. So just think of like a repressor protein. If I make a mutation in a repressor, typically it's repressing a process. I can remove that repression and then allow that process to continue on. Or I can make mutations in enhancer proteins where they gain a function and then they're able to push a process forward. You can also find informational suppressors or physiological suppressors. I'll give you some examples of those later on. And then suppressors, even within the same gene, sometimes you can get intragenic suppressors. Or if you're looking at things like a reverse genetics, you're always looking at extragenic mutations typically, where you're targeting genes other than the normal mutant gene. So true revertants, we could see these in the wild where you have a mutant and it can actually change back to a normal wild type mutation. Or you can have something in the secondary mutation in the gene that might help the protein form things, or you could have second sites. So let's take a look at some examples here. Again, forward mutations. It's where you're making mutations in a wild type and looking at mutants. Reverse mutations is where the mutant reverts back to a normal. These are not the same things as forward and reverse genetics. So these are just how mutations can occur. Here's an example of an intragenic. So our wild type is a leucine. We mutate it to a phenylalanine. It can actually revert back to leucine under some instances. So that's where you can get the same mutation in the same gene. We're going to skip that question there. Here's one where it's a partial revertant. So we've mutated to phenylalanine and it mutates back to isoleucine. And this is kind of where the genetic code has some redundancy in it. Isoleucine is a non-polar amino acid like leucine, and it could give you partial function in that protein. Here's an example of a second site mutation within the same gene. So let's say maybe your first mutation ruins an alpha helix in the protein. You could possibly have a second mutation somewhere else that could then allow that protein to at least slightly fold somewhat correctly and then give it some function. So there can be examples of that as well. What other suppressors are kind of interesting? I'm going to give you two examples of nonsense and smog suppressors. A nonsense suppressor, so this is an informational suppressor. So here, it's kind of interesting. They found a bacteriophage. So phages are viruses that infect E. coli or bacteria. They actually found a phage that would only grow in certain E. coli strains. So not all E. coli strains. It's kind of interesting. Phages, normally they form a plaque in the plate where they take over an E. coli and basically burst out like the Aliens movies, and then the phages infect more bacteria nearby. So you see a clearing zone, and they could follow the growth of these phages through those. And what the mutations they found in this phage, they actually had it concerned the head or coat proteins in the virus. And this guy, John Weiberg, found these, that the bacteriophage has a whole bunch of nonsense codons in the head protein, which, you know, we learned the nonsense mutations are bad. You're not supposed to get fully functional wild type protein from these kind of genes. And yet they found that the phage in certain strains could make it. So in wild type, this particular phage wasn't able to form a full head protein for the virus, but on a certain strain of E. coli, it could. So you're thinking, wow, how is the phage able to make fully functional protein, but only under certain conditions? It's really, really weird. How do you bypass a stop codon? Well, it has to do with the tRNA. So what they found is there was actually suppressor mutations in the E. coli. So an E. coli tRNA that was changed that actually helped the bacteriophage grow and make the full length protein. And what they found, that mutation was in the anticodon. It was in the anticodon of this tRNA, which then suppressed the stop codons in the bacteriophage RNA. Isn't that weird? So this is a suppressor. It suppresses the defect that you find in the phage. And it seems weird. Why would the E. coli do this? Well, E. coli probably didn't. But it was a mutation in that tRNA which allowed that protein to be expressed. And if you can imagine, there's different stop codons. So you could have different suppressors, and not all suppressors would cover all of the stop codons. So that's pretty interesting. You can get different suppressors for different situations. That's kind of what that's talking about. The smog suppression they found in C. elegans. These are also informational suppressors. And what they found here is that when you make mutations that could actually stabilize mRNA. So looking at the myosin gene, which is involved in movement, if you mutate the myosin gene, then you get animals that are paralyzed. And they looked at two different mutations. You have a nonsense mutation in the second exon, and then you had a deletion in the 3' UTR. As we know, the UTRs are really important for information or transcriptional control. So what did they do? They did a reverse genetics experiment. So you take a mutant. Well, I guess this is a forward. I take it back. Sorry. It's a forward genetics, but you start with a mutant. So you start with paralyzed animals and you treat them with EMS. So again, this is just targeting any unknown DNA, right? So you're just doing a blatant targeting of the DNA. And what do you look for? It's a suppressor screen. So you're looking for something that suppresses the paralyzed phenotype. What does that mean? Well, instead of just paralyzed worms, you look for worms that move. And maybe they don't move as good as the wild type, but they could be partial movement. So that's pretty easy. I don't know if any screen's easy, but at least looking at the plates, all you're looking for are worms that are moving. And then that tells you you have mutated something else, which is now suppressing that paralyzed phenotype. So what did they find? What did they find? Well, in looking at the RNAs, it didn't suppress the deletion. I'm sorry, the nonsense mutation. We wouldn't see any particular mRNA from that. But they found that the deletion of the 3' end you could actually see mRNA produced from that. And that's surprising. Normally, these would not be stable if you have a mutation in the 3' UTR. They're not stable mRNAs, and so you wouldn't get any mRNA from that. So if it is unstable, it's going to be degraded. Again, this doesn't suppress the nonsense, but look at this when they made these mutations. And so they found these proteins, they call them SMOG, SMGs. They found that these suppressor mutations gave you functional mRNA, which suppressed the paralyzed phenotype. And what they found in the SMOG proteins were actually part of the mRNA degradation process. So this goes back to our idea that there's a surveillance system for mRNA to make sure they're stable. If you have an unstable RNA, they're either going to be degraded from the poly A tail or from the cap and destroyed, or they're going to be cut in the middle and destroyed. You're not going to get protein from that. The SMOG genes were part of this surveillance system known as nonsense-mediated decay. So if you had a premature stop codon or an unstable mRNA, they're supposed to be degraded. Well, what happens when you go and you mutate the surveillance system? That's the SMOG suppressors. Well, you mutate them. Now the mRNAs that are typically going to be not stable, they're not going to be degraded anymore. If they're not degraded, then they can be translated, which means you can get some of the myosin protein from that. So this, again, was suppressing the mutations, but this is kind of a general suppressor. The SMOG genes probably, they look at all mRNAs. So this could suppress a lot of different mutations, not just the myosin mutations. So this is how they identified that surveillance system. Sometimes, if you think about it, if you just mutate the surveillance system by itself, you may not see any kind of phenotype because, well, the mRNAs are just being translated. You don't see anything. So here, you wouldn't have seen the SMOG phenotype unless you did it in conjunction with another mutation, which actually showed you that you're messing something up. The surveillance system has a role. So it's really kind of an interesting process there. Okay, so what's the point here? Well, again, we want to add this to our toolbox. If you're looking for mutations of interest, you can use forward genetics or forward screens to look for random mutations and then look for interesting phenotypes. Or you can target genes specifically using reverse genetics and RNAi. Here we want to take a look at a particular particular experiment done by Faye and Polly. It's going to be our next paper that we're going to take a look at. They used a suppressor screen and RNAi to try to find processes that would affect the function of these two transcription factors, Lin and SLR2. So that's kind of what they were looking at. It's a little weird because they started with a double mutant. And how it's written in C. elegans is you use lowercase letters. When you made this double mutant in the worms, they wouldn't grow past the L1 stage. So we have a growth defect. Worms go through 4 growth stages, 1, 2, 3, 4, and then become adults and then lay eggs to form new worms. So if you have a growth defect in L1, you're never going to be able to work with these worms because they're not fertile, they don't produce offspring, you can't keep them stabilized, okay? So what they wanted to do, what Polly and Faye wanted to do, well, they wanted to look for extragenic mutations. So here they didn't really want to do EMS because you could make random mutations all over. You wanted to use RNAi. And what's cool here is you can buy a library of RNA interference plasmids. And so they could target almost every single gene in the genome in C. elegans. And you can buy these. You can buy these libraries to do that. So they wanted to try to find suppressors using RNAi that would either be in the same pathway or in other pathways that would affect the growth that SLR and Lin are involved in. So remember, these are transcription factors. Transcription factors are going to be involved in the correct controlling of transcription. If you knock them out, you can imagine you can affect lots of downstream genes. They're of transcription. And if you affect those, that's going to mess up the worms. Obviously, the worms were not able to grow. So there's a developmental problem here, okay? So the idea, if we look at this side over here, genetic suppression, got to remember, not every transcription factor or pathway has a positive effect on a process or a pathway or a gene. You can have things that have both activation or repression, right? And they're going to be antagonistic. So here, if you look at the wild type worm, process Q is a positively affecting process X, whatever that is. And process S, again, these could be genes or proteins or pathways. It could have a negative effect, and they work together to control these processes perfectly. So you get perfect expression of whatever proteins or processes you're looking at, and you get the normal phenotype. Well, if I knock out the positive side of it, I could see a negative phenotype from that because now I've lost my activators or whatever process is helping that. And I'm only left with the suppression or the repression of that process. Well, what happens if I can remove that repression? I can suppress the disease phenotype. And knocking these each out, you may or may not see a phenotype. So knocking them both out together we can gain information about how they affect this process X, whatever it is. So just think of X as development. We're affecting development here. We want to try to downregulate other genes, suppress other genes or pathways to get some information on whatever development process this is. So how did Faye and Polly do this? Well, they started with the double mutant, but what they had in this double mutant is they had something that would actually help the worms grow. So it's an artificial chromosome that had a wild type copy of Lin35 on it and a copy of GFP. So just think of it as an artificial chromosome, really is what it is. They call it an extra chromosomal array. So it's a piece of DNA that's in the cell that helps to rescue the phenotype. So these double mutants, since they had a wild type copy of Lin35, then they would grow and they would glow green. So we would know they're the rescued worm. And you could work with these, right? These worms would grow up just fine, would give off baby worms, which would be green, and we could maintain the strain that way. Well, what's interesting about extra chromosomal arrays, about 30% of the time during meiosis, they lose the array. Well, if you lose the array, then you just have the double mutant again, and those worms are dead. They die at L1s, they have that growth defect. So this gave Polly and Faye a way to look for suppressor mutations. So what did they do? Is they took the green worms. So the green worms give off green worms, or they give off non-green worms, right? So either green or non-green. If they're green, we know they have the chromosome. If they're non-green, we know they don't have the chromosome, and they're supposed to die. So what do? What did they do? They used RNAi. So they fed RNA interference plasmids to each of these individually, to each of these worms, 17,000 of them, one at a time. You're knocking down genes one at a time. And what are we looking for? We're looking for any kind of non-green worm that survives past that normal growth defect. So non-greens that, I'll just say, grow better. They don't necessarily get to adulthood all the time, but they get past the L1 stage. And so that tells you that, okay, they're double mutant, but now we're doing something. We're suppressing something which is helping them grow beyond that mutant phenotype. So we're suppressing mutant phenotype. And again, it's going to give us information about what kind of genes could be interacting with slur and Lin, not just genes, but processes, pathways, however you want to think about it. What kind of things that could be involved in these transcription factors simply by looking for non-green worms that grow better. So again, it's kind of backwards. You're starting with a mutant phenotype, you're knocking down known genes, and you're asking the question, by knocking down which of these genes, which ones actually help our process, help our growth defect. And what did they find? They actually found certain groups. They found only like 27 or something mutants, 27 RNAi mutants, which helped the process. They found ones involved in ribosome biogenesis, prohibitins, and these SynMov, which a lot of them are involved in chromatin remodeling. Wow. So this was kind of cool, actually, because then they had to back up and go, well, how would tamping down on ribosomes help? That's translation, right? Aren't we actually hurting translation? How does tamping down on prohibitins, which are involved in fat storage, how would that help? How would tamping down on chromatin remodeling complexes help these worms? So when we discuss the paper in class, we're going to talk about how, well, remember, I'll just give you a hint. Like with translation, if we don't have our transcription factors there, transcription could be messed up, which also means that translation could be messed up. And so if translation is out of whack, if we actually hurt the translation process, maybe that brings it a little bit under more control, which allows these worms to grow better. So wow, we can actually hurt translation and help the worms survive better because they're all just messed up. And you learn a lot if you go back and look at these things. They're involved in nutrition and the digestion, that kind of thing, the development of the worm. So it kind of makes sense that if we can get it a little bit under more control, maybe we can help the worms survive. The SynMovs are not very discussed very well in the paper, but just think back to our chromatin remodeling. If we affect that process, could we affect transcription? Could we affect translation that way? Maybe there's a way that we can help them that way. And then the fat storage one, that just basically helps the worms use fat more. So what I want you to know here, first of all, how is screening important? what kind of information can we get from it, and what kind of mutants can we expect? So think back to the revertants or other mutations, second site mutations, or extragenic mutations. You know, why does mutating one pathway and then mutating a separate pathway, why is that typically an enhancer mutant? Because we're hurting multiple pathways that could affect the process that we're looking at. You also want to know how we can use RNA interference to do these screens. So EMS is one way you can make mutants and look for phenotypes of interest. Remember, RNA interference, we know what genes that we're targeting based on sequence. And then for Polly and Faye, we're going to talk about what do these genes tell us about their process affecting slur and lin. And then with these figures, they actually went back and double checked some of these. Remember, these are knockdowns. You can also double check them doing knockouts as well to see if you get the same thing. That's important. So hopefully that helps.